This week, we are talking about Camelina breeding and trait development with Julia and Anthony. Welcome to FGI Fridays. I want to start, as always, by acknowledging our funders, the Environment and Natural Resources Trust Fund, Minnesota Department of Agriculture, Clean Water, Land and Legacy Amendment, Builders Vision, Philanthropies, McKnight Foundation, among other generous funders that make this work possible. Reminder to folks, we're going to post this online. So if you're online, if you do not want to have your face shown, keep your video off. And I think we can turn to announcements. Mitch, do you have any announcements?
A little bit. So this is going to come out of the newsletter soon, but I want everyone to know where we are in our search for a new co-director. So as you all know, Nick Jordan is going to go into paid retirement in the fall, and we're bringing on a new co-director. There'll be a little bit of overlap there over the summer. We are going to be holding public discussions, or if you're familiar with academic hiring processes, kind of like a chalk talk, informal, very interactive opportunities for our candidates to have a conversation with our wider community. So anybody who's here at the U, who's engaged, our NGO partners, farmer partners, industry partners, the co-director role is pretty outward facing. So we wanted to have an opportunity for them to kind of embody that role for a minute and show how that goes. And those are going to be happening not next week, but the week after. So the newsletter today will tell you the times. We aren't yet ready to announce the candidates, but hold those times, please. They're at the end of the day. Hopefully you can work it into your schedule. And if you can only come to one or two of the three, that's fine. We get that people are busy, but we will really be looking for feedback. There'll be a feedback form. The search committee will review that feedback in significant detail. So your participation, the questions you ask, the harder the question, the better. So, we really want people to be engaging in the right choice on the next co-director.
I'm Julia Zhang. I'm the winter Camelina breeder. I'm also leading the pennycress breeding program. I'll talk about pennycress breeding in May. Today's me and Matthew will give update on the current status of the Camelina breeding. So me and Matthew will just be interactive, and I'll talk part of it. We'll talk a part of it, and I have to go. Okay. I'll start with the talk about the makeup of the team. This is the Camelina breeding, winter Camelina breeding. With the Forever Green was started in 2015. It was Matthew Alt's research graduate school research project, and over time we developed into a real breeding program. And then I became a breeder last year. Those are the people in the team. And basically right now, Matthew is an assistant breeder. If you see, we have a field technician. Oswaldo, he's 50% on Camelina, 50% on pennycress. And you see the Chris here. He was a technician on for Cargill and then a grant, but he helped a lot in the field also during busy time. Right now, he's back to be a grad student. And then the other two technicians are Sarah and Serena. They are pennycress. But with me on board, we are integrating the whole oil team into one. This is Matthew, and also we are embedded in the spring wheat breeding program. Got a lot of help from the other crops too. There, actually, the person in the cabin is Susan. Next one. Last year, our breeding trials was planted in two states. One, the first, most of our locations are in the southeast Minnesota into the sea, Waseca, Lamberton, and Morris, and then close by the home locations are the St. Paul, Rosemont, Waseca, Rosemont, and this is Becker. Further north is Grand Rapids, and then the out of state is is Carrington in North Dakota. Totally, we planted 8 locations. The major agronomic issue last year we encountered, which is the number one issue was a surprise was the winter injury, because the snow cover was terrible last year, and in the southern location, some the most severe winter damage was like 100%. We lost some plots, but in the north, it was interesting in the Grand Rapids and even in Carrington, there was no winter kill, 100% survival, and so that's kind of sad to see some of lines promising was had a severe winter damage, but it was promising to see in the northern locations, particularly in Carrington and winter Camelina did much better than the winter with the breeding materials by the NDSU program. So I think winter Camelina still have a future here in our area. Another big issue we had was as a whole of Camelina is the lodging issue. I met some agronomists had experience in Montana, even said the lodging issue. And I think there were two. One issue was caused by the root damage by the weevil. Another is the wind issue. And then in the southern locations, there was some sterility problem, like in Waseca. In June, the average sterility was 30%, and the shattering was pretty severe in North Dakota. And then I look at Jewel, the average shattering score was like 20% in North Dakota. Even in Waseca was like 10%. In some lines, you could see here could rise into 90 to 70%. I think breeding for shattering resistance in winter Camelina is a main breeding target here. As far as the disease and the pests we encountered last year, I mentioned that weevil was an issue. I worked with the disease clinic, and Brad did sequence the DNA sequencing of the sample we collected, and then there are two possible species, and then but they have 90. One is called cabbage leaf weevil, another is shepherd purse weevil. But DNA sequence could not separate those two species, only with dual phenotyping. So here is I have quoted Dr. Robert Koch. He was an entomologist. He said he went to the field. He wrote me back. I visited the plot on campus with Julia. Well, some of the plots are really getting wet. Other problem we saw is the ester yellow was very prominent. In some location, you see the pods are purplish. And then the damage by the ester yellow depends on when the infection started. I'm working with the virologist, Dr. Robert Alvazo Pinto, to try to sequence that to decide the disease instance, and also the most ultimate goal is find the vector and how to control the spread of this problem. Bacterial leaf blight, if you is obvious because we got a lot of rain in June, and then you're working with Dr. Davinci, the bacteriologist, to ideate. And also powdery mildew was pretty bad in the late season, but I don't think there's a damage on the yield. Based on what we have seen in the field, talk to the agronomist, I summarize our top rating objective would be certainly the yield. But you have to remember, and then the yield is relatively negatively correlated with maturity. Early maturing material tend to have a lower yield, and as a winter crop, we have to select early materials for the second crop. There is a balance of yield for maturity. The top 5 agronomic priorities. Number 1 would be winter hardiness, and we are using the field data to do selection to discard anything susceptible else also. And thinking to do a greenhouse free or grow chamber freeze test to discard the susceptible material in the early generation. And the next one get is early maturity and resistance to lodging and resistance shattering. I I talk about and how to manage the weevil issue. Can we, maybe there's a way you can do just treat the seed with the coating and don't have to select for the resistant ones. And the seed size we hire a lot from our industry partners to get a bigger seed size, the material for the crushing. Easiness to in the crushing plant, so they don't have to retrofit just for this Camelina. As far as the seed quality and the need to increase the oil yield, and certainly the meal is got to be nutritious for the animals. Would be the main component will be protein, higher protein, lower fiber, and low glucose mix.
Hey, Julia.
Yes.
What was the reason for the sterility that you mentioned?
I think it's the heat tolerance issue.
Yeah.
And then maybe Camelina is susceptible to the heat, to extremely
Was it
high temperature.
was it just a few genotypes or?
I noted was I think there's a difference in the genotype. Some of them is more resistant to the more tolerant to the heat. There's opportunity for selection. It's I didn't see this problem in pancreas. Oh, here's the picture. This is our Camelina. Look really nice in the. I like to show this. This is the keratin and tall and very healthy. And then in the same time, this keratin our Camelina field. This is the winter. In the breeding trial, you see that all those holes was those winter wheat was killed in the winter, and then the space was filled by the wheat. So the promising the winter hardiness on the Camelina is better than winter wheat. In winter, Camelina is better than winter wheat. This is summary report of the yield location performance of the different locations. You can see the listed 8 locations. We planted the further north location, Grand Rapids, before September 1st, September 15th, and then our last planting location was in the September 26th. And the flowering, the earliest flowering was happening in St. Paul started in mid-April last year, and then mature early was in the mid-June, June 18th, and we were able to harvest the St. Paul location before July 4th weekend. The last week, most of the location was harvested after 4th of July weekend before in the second week of July. That's what the maturity range of our existing germ pressure. You can see the winter kill I talked about here, Carrington and Grand Rapids, 0. And the St. Paul's maximum was 100. And then the yield, Carrington, surprisingly, this is the first year we were there, the yield was pretty impressive. It's similar, a little bit higher than the St. Paul. I think the statistics is over about 1,000 kilogram per hectare. This is the average of the location, is not by not genotype.
How does the Carrington yield compare to Pennycrest at Carrington last year?
I think it was similar to the Pennycrest. Some lines is higher than the Pennycrest. Yeah, it's I think one person commented Camelina has a very high harvest index. It's amazing that a short plant can generate such a high yield. Pennycrest is even taller. The harvest index is not as high as Camelina.
Yeah, these average yields.
Yeah, these are average yield, not the top. And Grand Rapids was pretty nice. It's just about 200 kilograms less than or considering for far north it is. And then Becker, we learned Becker location was irrigated and it's still terrible. Because some of plots have handled winter killed, but winter just suppressed the yield. Still have just too sandy for growing Camelina, I think. And then Rosemont Wasica was not as good as some location. And Lambert and Morris didn't have data, just those locations didn't have the irrigation. If you remember how dry the fall 2024 season was, and we just couldn't walk away from those locations. This is a correlation of talking about the correlation of the seed yield between different locations. What was very interesting was since the correlation of the yield in the St. Paul with the other location was pretty low. You look at that at about 20% with every location. And then but the correlation of the Grand Rapids with carotene was pretty high. That explains as further north is cooler environment. But even Wasika and Rosemont correlation among all the rest of locations was like 60 to 70 to 80%. So I'm waiting for this year's data to confirm maybe how representative the yield, how useful the yield trial is for location would be to us in the breeding program.
Sorry, Julia, I'm just wondering, if I were to read this, all I would see is that the yields in a certain location are X, but I'm not sure what is being correlated with location and yield.
But no, this is the trait location, right? Correlation of yield among different locations. Is that answer your question?
It's showing that yields are kind of similar between Carrington and Grand Rapids, for instance, but not similar. They don't follow a similar trend between Carrington and St. Paul.
Right. That's what I mean. Yeah, if you have a high yield in a line is high yield in St. Paul, it's not necessarily high in other locations. But if it's high in North Dakota, Carrington, most likely will be high in other locations like Grand Rapids, Rosemont, and Wasika. So I'm just this whole idea is trying to see what each location data is telling me how to use the data. That's why we needed to do a multiple location test, stability of this trait in different environments. This is a little bit busy table, and then we are just look at the heritability of some of the traits using last year's data, and then the first two. I just pointed the first two locations. First is a seed yield. The carotene, the heritability is lower than the seed yield. The heritability in St. Paul is 0.7. And then for the oil content, Grand Rapids is 0.6, but it's 0.3, but in carotene it is relatively high, and then St. Paul is 0.6 or 0.5. And don't have to worry about the statistics criteria to interpret how accurate this heritability estimate is. Last year the CMA 2207 is the line Matthew developed. It was used as a candidate for variety release, so variety release candidate. So last year we grow it in multiple locations to see its performance. This is the result. It was growing in 5 locations in Minnesota. We see that, and 1 location in North Dakota. And also we have a partner, industry partner, grow this line along their line in Napa, Idaho. And then, but overall, that's yield it was not as high as Jewel. Jewel is checked on this table. And but this line mature certainly flowered 1 week earlier than Jewel and also matured 4 days earlier than Jewel. Certainly the early maturity is real, but the oil yield, which is a combination of oil and yield, is less than the Jewel, the check, and the seed yield. Jewel is best, the oil content was best. So, and overall, this line was in the Minnesota and North Dakota locations, data was not as good as Jewel. So we, based on this data, we decided not to release this line last year. But we are still happy to see in Idaho this line compare with the winter Camelina lines developed by a private company breeding our line. The seed yield in this one, our CMA 1007 was 37,000 kilogram per hectare, but the private company's line, their highest yielding line was 2,800. We yielded 900 kilogram per hectare. Then private companies. But and also this line was mature earlier than this private company's line. So this we haven't given up. CMA 2207 in last fall we planted keep it in our yield trial to see how it performs. Oh, this year's data. When we'll see how the data after harvesting in July, how the data look like on CMA 2207. So this is a chart, the multiple site. This is our PYT human yield file, oil yield content by genotype. Those are the breeding lines. Those 19X, CMA 2207. I mentioned that our most promising breeding line Jewel check. The other lines are also checks. You can see some of them, most of them, this Jewel still pretty good, very high. Our line didn't perform, was similar or lower than the Jewel, but a few of them went to this year's replicated trial just because the winter hardness was good and the maturity was very similar to Jewel. So 1 to 6 lines went to this year's replicated trial. 3 of them, one of them was given to I think Joseph have it. This one, the 21 or 21X, this line, but is in the 7 location trial growing in 3 different states. And 3 locations we thought is still promising because it is earlier, we put it in a 3 location multi-rep trial. So that's the performance of our most advanced materials.
But it's that winter line too that looks like it's
Which one?
the one that has a very tight distribution and it's at the top.
Yes, we do it.
Yeah, winter two,
Winter
it's in
two.
3 locations.
In 3 locations.
We put it in 3 locations.
It looks like in this year it was the best.
It was only in 1 location though.
So
I see.
Yeah.
but that's why we were cautious.
Yeah.
We didn't put it in the 3 locations.
Are those
7.
on the right, those 4 are those are reading lines or are they, you said maybe they're checks?
I think they can be breeding lines and we are testing them. I think if they are good, why not release it? Okay. Yeah, and also I'm breeding heavily with them too as a elite breeding parent. For 2025 year trial, we think this is next set of materials will have better winter hardness because they were selected on the extreme winter cold pressure. And if you look at this and also we first selected the winter hardness. After that, all the plants, single plants harvested the winter through the margin and select it for seed size. You can see there's a range of variation of the seed size. We just pick the top part of that. Those went to the further testing, the yield trial. And then total number of entries in yield trial was three times higher than last testing cycle. And then those were planted in 3 locations. With the four locations, one of them was 4 locations. We also jumped some lines. We didn't go to yield trial. We just didn't go through any yield trial. Jump to the 4 location test just because. Just in the nursery agronomy make it look good. And then see, this is the seed size. Matthew put a lot of effort to select a higher, bigger seed size, and last year also collected green percentage green per plot, and we did a correlation between the green percent, the green area, and with the southern green weight or X. This percent green, there's a positive correlation between seed size or southern green weight and then the percentage of green in the plot. So that my real belief is correlated in the yield or not. At least for the eco service, that's the good news, right? In this winter, in the fall, we do want to have a bigger area of with the green cover can reduce the soil erosion. In the major challenge of the winter, then in the winter Camelina breeding, number one is there's a very limited number of winter type germplasm, similar to winter pea or worse, And in the first few years, and Matthew spent a lot of effort to collect and identify which one is truly winter type. So far, he only found 15 winter accessions in the gene bank collections. Those are from former Soviet Union and Poland, Canada, and the US. But there is a lot of spring Camelina with germplasm, and also we did a yield testing. The spring type have much higher yield than spring. So the strategy right now for us is just to make a winter by spring crosses to bring the high yield and genetic diversity from spring, but maintain the winter hardiness from the winter parent. And so because of the winter hardiness, and then we are still trying to understand how to utilize this precious amount, only 15 lines of those materials. So we are in the last fall, we did a study of the winter hardiness and the flowering habits of those winter lines, at least here. And those lines, you look at that, even 15, the Camelina sativa is the true the Camelina material we are talking about. And this 40 chromosome, even with there's other ones, there's another one is 12, 24. 38 chromosome. This one is Camelina microcappa. There's also, those are all hexaploid. There's a tetraploid, two lines are different species. Looking for opportunity to do white cross from these few lines. But Matthew will talk about what we found out in the winter hardened winterlization study.
This was an experiment that Julia said that, yeah, we should check it out in the greenhouse this winter to just see kind of what the if there is a vernalization requirement, and if so, what it is, for 2 reasons. First reason is maybe we can shorten the breeding cycle. Are we vernalizing too long, or maybe we're not vernalizing long enough? Maybe we could vernalize longer but then shorten the breeding cycle still. But what it turned out is that we were maybe vernalizing too long. We were typically for 3 weeks, but the data shows that the first days to first flower at 2 weeks of vernalization time got, I mean, that was the shortest days to flower for 2 weeks vernalization time. So 58 days to flower for 2 weeks vernalization time. That's average across all these lines. So some of the actually lines in the blue boxes are different species of Camelina, and then the gold box is also different species, but I'll get into the gold box in a second. But the other species of Camelina have a higher vernalization requirement, the first two here. So for most lines, 2 weeks is probably optimal for most of our breeding lines too. So shortening the breeding cycle, if we take off a week over a year, that's 3 weeks off of the breeding cycle, because we can typically fit 3 cycles in a year. So yeah, just trying to move as fast as we can.
Matt, the ones without an error bar, does that mean they didn't flower?
The ones without an error bar just didn't have any error.
So zero vernalization.
Yeah, so look at the asterisk. 68 at burn 0 weeks specifies never flowered in experimental time frame. I just didn't want to go to 100. I mean, we didn't want to have, so 68 days was the time frame. We didn't want to drag this experiment out for months and months. So we cut it off at 68 days. The reason it's going beyond 68 days at week 1 is because I add 7 days to that 68. So it goes, the highest there is 75. And then in 2 weeks, I had 14 days to that 68.
Okay, so I was wondering why it would go up at the end of the curve without explaining.
Yeah, so the next figure shows like, okay, now this is post-fertilization. So post-fertilization here, that's the other question. What lines, post-fertilization, what's the fastest to flower by genotype? That's another thing we're interested in. Our facultative line without any fertilization flowers pretty fast still, with 0 weeks of vernalization. And then we have a bunch of lines, winter lines here, that don't absolutely require vernalization to flower. They flower a lot slower though than our facultative line. So in a sense, these are kind of facultative lines too, in a way, but they just flower more slowly than our early flowering facultative line, I guess. So yeah, that's helpful to know, that, yeah, we might have some more kind of sources of this facultative trait, little variation there. So also this gold thing now comes in. So yeah, that's a different species. And at first it was kind of, it only required 1 week of vernalization time. But then the reason I highlight it is because its overall days to flowering, even after 4 weeks of vernalization, it's just more slowly to flower for that other species in the gold. So another observation, and yeah.
That's a strange thing. Look at that one. So what happened was the 6 week vernalization flowered later than the 5th vernalization. It happened across different genotypes. Look at that line. Is that a question mark?
Yeah.
That's a very weird behavior. We are going to repeat it.
Yeah.
If you go back to the first one slide, that's the same thing, right? Go back.
Go back the slide.
Yeah, so you see because the 5 week, 6 week, look at the 7, 6 week is longer because we started from the day 0. This is equalized. Yeah, look at that. Every one of them delayed. If you keep putting in the 4 week longer, it'll be fun if we do another week, right? 7 weeks.
Yeah.
To see if it's longer than 7, 6 weeks. We don't know if it's a issue with the bench or this is a behavior of winter Camelina in the species.
Mm-hmm.
That's strange. So we put a question mark.
Getting into some growth chamber data here. This is from an experiment I did in 2021. There were vernalization treatments, 3 weeks or no vernalization. And then the day length treatments of 21 hours and 5 hours. So going really extreme long and really extreme short days. Partly because 8 and 16 was already tested, so I wanted to do something new that hasn't been done. So yeah, under long days with vernalization, even at 5 hours, one of our winter lines here flowered. So there's an error bar there. Joel actually didn't flower, so maybe it's got a day length response, Joel. But this winter line would flower under short days. So that was interesting. And then all of our facultative, these are all facultative, different facultative types over here. They all flowered under short days as well. So Camelina perhaps could be day neutral. That's kind of what I want to highlight from this, at least for now. And Jim actually thought, oh, we should do a temperature treatment. So we coordinated with Cargill to do a temperature treatment as well. And this is what that kind of experimental design looked like. So these are all the temperature treatments, and it's mimicking like what they used for their data to mimic was Wasika past 5-year average. So they got weather data from Wasika past 5 years and averaged it. So this is like an average fall in Wasika, and then mimicked September 20th, 30th, and October 10th planting dates using these 4 lines of Camelina here, one of them being a spring type Camelina, and then we have our facultative Camelina here. No vernalization. So there's no vernalization treatment here. They just, nothing got vernalized. We wanted to see, partly the motivation of this was like fall 2024 was a super warm fall. And so we had some flowering of our facultative line in the fall last year. We're wondering, okay, maybe day length, if we change our planting date, maybe day length can suppress that flowering so it doesn't flower in the fall. And that is kind of the result we got. We were kind of hoping that the September 20th treatment would have like flowered, kind of like it flowered in the fall of 2024. But since we used the average temps, it probably wasn't warm enough to make that treatment flower. So if we do this again, we'll just use that 1 year, that warm year for the data. But what we can take away from this is in a 5-year average normal year in Wasika, Minnesota, winter and facultative would not transition to the floral meristem when planted on September 20th, but the spring type would. This is the spring type. And you can see it bolting. So at least we can see that our facultative is different than spring. It is not a spring type. It's a true facultative. So that's probably why it does better than spring types when planted in the fall. We've tried planting spring types in the fall to see what happens, and it's not good. So yeah, the facultative type does better than spring types when planted in the fall. And yeah, planting date can be something that it's a lever we can pull to try to optimize the system and make sure it doesn't transition to floral meristem in the fall, because that takes a big yield hit when it does that. So
what's the cross between like the facultative in the spring and the facultative in the winter? What have you crossed those, or like what's the behavior of those plants?
So the facultative parent, so CMN 2207 is a progeny of a facultative by winter.
Okay.
And selected for early flowering from that cross. So yeah, so it is a progeny of that cross. But I mean, you know, it segregates. Basically, we know that CMN 2207 doesn't have winter type FLC gene, which controls the vernalization response. So it's spring type in that regard, but there's some mysterious thing that we're still trying to look into of what makes it different than spring types. We still don't know what that is. So yeah, it's been a tricky one to get to the bottom of. But Possibly, yeah, this might kind of point to day length. So, yeah, I think that's all I've got.
That's one. I guess.
Yep.
And then currently, I think the Camelina breeding program is expanding, and then it's mainly realized by the cardio help. And then there's a research facility. We have access to the research facility in Fort Collins, and then so we also I changed the breeding strategy after I saw the severe winter damage on our crosses. Right now, the new crosses are still make up some of the winter by winter crosses. We haven't tested yet. Another strategy is doing the winter by winter. Most of crosses what we are doing winter by spring. I'm worried that the spring typing too much, we will lose a lot of lines. We won't have the winter hardness just by winter. So I'm making 3 crosses. The first cross winter 1 by winter spring, and then make another step across the winter 2 to bring some genetic diversity and from spring type and reinforce of the winter hardness. And because cargill's help in the advanced generations and increase the seed, and we in the fourth 22, 26 planting will maximize our trialing capacity. I think I feel the limit of our resources now. That's it. This is a picture is how good our plot look like because we got a new planter.
Has anybody looked at pennycress or camelina since had these thaws?
Yeah,
I mean,
I took some pictures before the snow, the most recent snow. There's a mix of kind of tan and green for both camelina and pennycress. And the tan might come out of it.
The color?
Yeah, the color.
The purple?
Well, just more, I didn't see purple yet. There might be some. I didn't look super close, but just overall, like, in the pennycress field, Julie just got back from China, so everyone know that, yeah. So, update in the pennycress field, the lower spot has a little more tan.
Okay.
For pennycress, but there are some green spots in the lower spot too, so those ones might be good.
So the Camelina is funny. Last year, after the winter, the Grand Rapids, the leaf were purple. I said we are doing a maple tree breeding. I was so purple. I just recorded the shade of the color to see if it correlated with anything. Is a genetic difference or related to yield? We didn't find out anything. Yeah.
Yeah, so it might be a good selection year though, because we see it's not like 100% winter kill. There's green, there's a mix, so it's probably a good selection year.
Yeah, we'll see if we got it purple.
It looked pretty good from our picture this morning. And
Oh.
last week before the snow.
Okay.
Yeah, there's a low spot in the pennycress field where there's a little more like damage, it looks like, but from the edges are much better. Yeah.
Okay.
I would definitely watch out for those low spots, because when all that snow was melting so fast the first time, and it was getting cold, like, I wouldn't doubt that there was a little bit of peak shooting going on in various fields.
Yeah.
Yeah. Before I left for China, I walked by the Camelina field. I didn't see it. Camelina looks okay, but we got another round of snow, right? Right.
Yeah, there's some tan in the camelina too.
Okay.
So, yeah, but there's some green. So it's, yeah, good selection year.
So most of you probably know me from my work with pennycress mutant gene index that I've been using for the past few years. And about 2 years ago, Matthew reached out to me and introduced me to the Camelina mutant gene index creation project. So we're excited to show you the progress that we've made. So just a quick overview, this is all funded by the Cargill Philanthropic Gift, which means it does have to be released as a public resource. We're not at the point of releasing it yet, but we do have some excellent progress on it. And the end result of this is it's going to be a resource that will enable reverse genetic screening for specific traits that people are interested in. And because reverse versus forward, people always get these confused. I always throw the slide in here. Remember, reverse genetic is where you know the genotype of what you have that you're looking at, and you're working backwards to determine whether or not it has the phenotype that you're interested in. So the quick outline of what the creation looks like. All of this stuff is based on EMS mutagenesis. Very, very standard technique. It's been around for decades. And we grow out these M1 generation plants. We collect the leaf tissue, and then once they reach maturity, we harvest the seed and we store it. So we're sequencing the plant tissue. We got some of those results back actually just recently. And then we run it through a bioinformatics pipeline to turn that sequence data into something that can be easily used by just a technician or a researcher, as opposed to having to pour through tons of technical detail. So the idea is sort of this, we're making something that is easy to use for the end user. So we're enabling people. But all of this is going to be based on what the quality is of the things being fed into the system. So happy to report UMGC, we got our results back from them, and we have extremely high quality, nice sequencing depth, just below 10 across the genome, which is excellent. And the only thing that stood out was just a little bit of adapter trimming, and everything looked clean. So, you know, the old adage is garbage in, garbage out. So you got to make sure that your stuff going in is nice and high quality.
And shout out to Chris, because he made it so that there was not garbage going in.
Yes. So for a bioinformatics pipeline, the standard pipeline for anything when you're working with sequencing data is you get your sequencing data, you generate your alignments, and you create what's called a VCF file, which is a type of file that you can plug into almost any type of analysis program. It's very standardized. But in order to create a gene index, we had to add an additional pipeline. This is predicting the effect of the genetic variants. So there's a program called SNP-F that we use, and I had to create a custom database of the Camelina genome that basically determines, looks at the annotations, finds gene starts here, ends here, and this is what your reading frame looks like. And then we use that to create this bin file. It's just a binary that you feed in that plus the VCF, and you get an annotated VCF that gives you the impact. And then once we're done with that, I run it through a custom R script to turn it into something that is easy to distribute that you can just hand to another researcher. So this is an example of what it looks like. Shout out to Matthew for generating this. Each of these colors represents a different gene. You'll notice the two gigantic black columns there. For IP reasons, we had to redact that, but there would be specific information for what the nucleotide substitution is and what change that has on the amino acid structure of that protein, which could be a substitution, it could be an early stop, it could be any of those things, including splice sites. So this is a quick screen that Matthew did showing just a number of high impact for various traits of interest. And what you would do is you look at a giant spreadsheet that's like this, substantially larger, of course, and then you say, okay, I'm interested in oil content. So you reduce it down to ones that are just variants within the gene of interest. So in this case, this increased oil gene that will remain nameless for the moment. And these are all of the high impact variants that are present in that gene. You know, some of these you can see in the variant type, there's early stops, so it just terminates the gene early. Some of them are splices, so it affects the processing. But all of these could potentially disrupt the function of that gene. And these columns here are the ones that are generated by that program I mentioned before. This is the SNP-F stuff. So this is the actual functional impact that we're interested in. So you look at these in the full gene index, they're not all going to be high impact. So you can filter through these, determine which ones are worth spending your time on. And then you go look at this accession column here, and that just tells you which line to pull out of storage. And then once you've got those lines in hand, you just grow them out and you do phenotypic evaluation to see whether or not it has the trait of interest. And because these are relatively small pools of seed that we're working with, we also want to use that opportunity to increase the amount of seed for future use. So if you want to go out, you find something that looks promising in the small scale, and you want to go out and do an agronomic trial, you're going to need the additional seed. So the status of the index right now, that SNP-F database was constructed. And pipeline has been developed, and we ran a draft index for the first approximately 100 lines of sequencing data that we have. We have new sequencing data that is currently, as we speak, running on MSI, currently being processed. So we're going to be in the range of about 300 lines total. And thanks to the work that we've done so far, we were able to secure some Forever Green funding in the last round. and that's going to be used to add an additional 100 lines. So the next steps for this, we're going to grow these out, extract DNA, and do all the usual work to get those additional 100 lines added. And then we are going to continue the pipeline development, just to improve the overall quality of that final thing we're getting out to the users. And then we're going to validate that by successfully deploying it to identify traits. So just because we found something in this table doesn't mean it's the actual ground truth of what's going on biologically, so we need to verify it. So I want to acknowledge, of course, Cargill is the funding source for the work that we've been able to do so far. Extremely important, and that's that 300 lines that we've got running right now. And Forever Green for giving us the opportunity to expand on this resource and improve the strength, because the number of variants that you have within this table, it really determines the value of the gene index itself. And then I've mentioned Matthew before, but you're sort of the reason why I'm on this project, so thank you for that. And Chris and Serena did a bunch of the work to get us to this point, to get us our things submitted for the for sequencing. So thank you both for the contributions there. And a lot of this work was inspired by the work that Ratan did, creating the Pennycrest gene index. So we reverse engineered a bunch of stuff that he had been working on, but wanted to give him a shout out because a lot of the stuff that I've been doing has been figuring out that pipeline to use the gene index and get things filed for intellectual property. And everyone on that slide did a lot of contributions to this work.
Yeah, Serena did the first round before Chris started. So we've got the ones that you extract DNA on in there too. Yep. So,
So while you still have this one up, can you go back to I think it was your second Excel table where you had just the high oil? Do all of these represent mutations of the same gene, or are these all different genes in this example?
a lot of them there's a lot of overlap and gene families.
Okay.
Yeah.
I just noticed there did several different chromosomes represented. So
Yeah, and then hexaploidy, there's a lot of like the hexaploidy issue too. So that's why there's we get a lot more hits in Camelina.
So would this represent all of the high oil potential for the current MGI?
We also have, besides the high impact, we have like what's called moderate impact. So that's just amino acid substitutions. And some of those can be good too. But this is all the high impact, for that gene family and various genes, Mm-hmm.
And the gene families in this case were done through just identifying homology through BLAST. So they should be very similar, but there's a lot of complexity when you're dealing with something that's hexaploid.
And that's my question, and I don't really understand polyploids genetically, but would you? I know probably it depends on the trait and whether it's quantitative or qualitative, and how genes interact across those different genomes. In some cases, maybe we'll see a significant effect of just having it on one of the genomes, but in others we might need it on 2 or 3. Is that the case?
Yes.
Then is there a way to transfer it, get it onto the other ones? We know that's a key trait that we'd like to see across all of them.
I think for the type that we don't see a phenotype, even though it looks like a good knockout, stop codon. Like even this oil, for example, the one Anthony showed, we're getting hits in different subgenomes. And so we know what chromosomes are in what subgenome. And so we can make crosses. Like if we got a hit in this subgenome and we got a hit in this one, then we can make a cross to try to stack them in the different subgenomes.
Right. Yeah. Getting it to cross over to a different subgenome is a different story.
Right. So we have to make a cross, like a flower pollination cross.
Yeah. Mm-hmm. That makes sense. I'm just wondering if we don't happen to have hits on the same gene in multiple subgenomes.
Two we do, but
okay.
They're more rare. Yeah.
Sure.
Mm-hmm.
But another viable strategy is if you know that one, you have a hit on it, you could go in with CRISPR.
Yep.
Target the other members of that family.
Mm-hmm.
If there's enough homology.
Right.
Yeah, and for Pennycrest, we've been doing a lot of gene index plus CRISPR joint filing with Illinois. So using this does not rule out using CRISPR at all. In fact, they work very well together. So I do have the extra data I wanted to include. Yeah, so these were from the increased oil ones that we were just talking about. And in this case, Matthew went through and pulled out a bunch of lines, grew them, and we have, you can see our NIR readings for the oil content, so a quick estimation, and whether they're heterozygous or homozygous. So you can see here that we do have several lines that have increased oil content that are potentially worth pursuing in a larger scale, because this is a very small scale test. Maggie, what's the baseline for like Joel for oil content?
So in the field, Joel is around 40,
Right.
but in this cohort, that 39.96, I think is just it merits further testing.
Mm.
Yeah, because this is grown in like a grow chamber greenhouse. So yeah.
Mm.
So how do you explain the 24%?
That cohort had pretty small seed, actually. So I think it was greenhouse grown, and I think we should still grow that out again too, because I noticed that cohort of mutants, the seed was a little small, like maybe half or 3/4 of the size of normal seed. So I think it could be that maybe there's a background mutation and the seed is small. It could be that it just didn't grow well at that time, but I think we need further investigation into that 24% one.
Yeah, oil content is one of those traits that has a lot of things feeding into it. So it's difficult to nail down exactly why something was smaller like that. But I have actually seen counterintuitive results in pennycress where we thought something would increase oil content and it actually reduced it. So it is worth noting that this is just affecting the way that one gene functions, and the actual structural impact of that gene, it's very difficult to predict that type of stuff.
Plus, there's a bunch of background mutations that work
Yep.
as well, right?
Mm-hmm. Yeah.
Are you pursuing a near isogenic line approach? We talked about that, I want to say a year or two ago in this room.
Yeah.
Pennycress.
I guess we should get that back on the radar. Yeah.
Should any of these hats you can sell and get the two homozygous classes and compare them directly, so that should cancel out some of those background effects.
Right.
Yeah, that is going to be part of when I do validation. If we have something that we're looking at that's head, obviously we do the cross so that we can get that distinction. And that's what I've been doing with pennycress is, obviously you want to get that homozygous mutant. And if you have something that's head, it just takes an extra 4 or 5 months to be able to reach that point.
But those are most of those loss of mutation, loss of function mutation, right?
Mm-hmm. Yeah.
So if you do that, that's recessive. What do you get from self-impact? Because
you'll get-
but you have your 39, the 40% is homozygous already.
Yeah. So that one we wouldn't do the self on, but some of those other ones that are het.
Like maybe the 38.
Right, the 38.67. Yeah.
Right.
The purple and the light blue over there, those are the two lines. Well, purple is one line and light blue is one line, and they had hits in different subgenomes.
Okay.
So it's a little more rare, but it happens. Yeah.
Yeah, the other thing to keep in mind is that even if something here says that it stopped gained, if you're looking at something that's 1,000 amino acids long, if you gain a stop at 900 versus gaining a stop at 30, very, very big difference on what's going to happen biologically.
You mean the gene? The gene?
Yeah, in the position in the gene. If you just truncate the end of it, sometimes the gene is still functional.
Okay.
Or if there's a negative feedback recipient, or you can end up essentially turning off certain functions while leaving the rest functional. So it very much depends on the gene you're looking at, and you have to look at the annotations and the structures that exist in literature to be able to predict some of these things. But the takeaway is we got a bunch of interesting things going on for one trait, and we've got the seed on hand to poke around and see what happens.